Alrighty. All right, so let's go ahead and begin on the dot. So hello, everybody, and welcome back to MSA SPS, or the Mycological Society of America student and postdoc section. And this is the Students Mycological Symposium. So we'll be starting on the dot to give our speakers enough time to explain their work and for questions. And so we're really excited to have you all here for our third meeting. My name is Tanya, and I am a fourth-year microbiology graduate student in Dr. Jason Seich's lab, and I'm the vice chair of the MSA SPS. And here I have Maria, who's the webmaster. I have Jillian, who's a social media coordinator, and hopefully we get other members of our team on here as well. But first of all, welcome, right? I would like to say, if you have any questions, please go ahead and write them down in the chat and we can address it. Either that, or I can read it to the speakers, or I might call you and then you can read up your question to them. This meeting, again, I'll say it again, will be recorded and will be available on YouTube. We will be sharing the link through Facebook and Twitter. The talks will be about 20 to 25 minutes long, leaving about 5 minutes for questions, thus filling in the hour. So our two speakers are going to be from Dr. David Rizzo's lab. I just noticed he just signed on. So first, let's begin with Sebastian. So after completing his master's degree in Chile, Sebastian initiated his PhD in pathology in 2017 under the supervision of Dr. Dave Rizzo at UC Davis. Most of his work has focused on the impacts of Phytophthora pathogens in natural ecosystems, assessing patterns of distribution, diversity, and the potential risk these pathogens pose to native vegetation. So with that, Sebastian, let's begin. And you're muted. Okay, you're good.
Okay, yeah, that's the first thing. Okay, and now I'm gonna share my screen. Yes. Okay, cool. Okay, I think that looks okay. Well, first, thanks for the invitation. I haven't spoken in front of a crowd in a really long time, thanks to COVID. So it's good to be back and doing this kind of stuff. And today I wanted to present a little bit about the problems generally related with Phytophthora and restoration areas that were a lot of these problems that we're seeing in California, and how these pathogens are being introduced into natural ecosystems. And so I'm gonna generally talk about Phytophthora a little bit, the problems with restorations, and then dive in a little bit into the case study specifically in the Angeles National Forest, where my dissertation topic is basically focused on. So I'm gonna start off explaining why maybe I shouldn't be talking in a mycology student colloquium, because Phytophthora, technically they're not fungi. They fit into this group called the Ophiostocons. And these are like fungi, as we know, are more related to animals. And these Ophiostocons, this group is known to have a posterior flagella. So in some point of their life cycle, they have this flagella, which is the difference with omycetes that fit on the other side of this tree of life, where omycetes fit into the straminophyllae, or the heterocons that are known to have two flagellas. And omycetes are more related to brown algae. Now, if we dig deep a little bit into more of where omycetes fit, they fit into this super kingdom of Chroma violata, with organisms belonging to the Chromista, which in some point of their life had the ability to do photosynthesis, and then it was lost. And this is where we find the omycetes here in the lower part of this tree, in the group of the pseudofungi, but this probably is already changed or is going to change soon because these are updated very frequently. Now, if we dig into the omycetes, they are formed from by several orders. Some of the common known ones are Saprolegnias, commonly encountered in freshwater ecosystems, and they're actually known to cause disease in fishes, especially in stressed out fishes in nurseries. They're quite a big of a problem. But then we focus on the peronosporales and the pithiales, which are more soil-borne and associated with plants. In the case of peronosporales, is where we find the Phytophthora, and closely related to the downy mildews, these obligate plant parasites. So omycetes at the same time is a quite diverse class of microorganisms. Um, Now a little bit about this life cycle of Phytophthora. Phytophthora, and this is where it's kind of more similar to fungi, where it has a thallus and a mycelium, where in certain that it can go into an asexual sexual cycle depending on certain stimuli from the environment. From the mycelia, it can develop these asexual structures called sporangium, which can either dislodge and germinate and cause disease or infect plants, or it can open up and liberate zoospores between 15 or 20, depending on the species, and forming these swimming zoospores. These swimming zoospores then swim to through chemotaxis to either root material or some plant susceptible plant tissue and attach, insist, and then form a germ tube. And then with either entering through stomata or cell wall degrading enzymes, they're able to penetrate plant tissue and initiate disease. Now, again, depending on the environment, they can undergo a sexual cycle where where an ogonium is produced, a structure that holds the oospore, and then the male part, the antheridium, then fertilizes and creates an oospore. This oospore is also known as a resistant structure. It allows them to survive in adverse environmental conditions. Then from this, oospore can germinate and again go into the asexual cycle, producing sporangiums, most oospores. There's also another asexual resistant structure that are known as chlamydospores. Oospores and chlamydospores is what allows Phytophthora to survive extensive drought periods or stressful conditions. Now, Phytophthora can also cause disease in different ways, depending on the host, depending on the species. Some species are known to cause root rots, crown rots, like as we can see an image up here. In nurseries, also with seedlings, it's a big problem causing damping off, where the seedling is strangled and collapsed. And then there's also Phytophthora, more aerial Phytophthora that affect aerial parts of the plant, like leaves or stems, causing either leaf blights, leaf necrosis, or it can cause and infect stems of adult trees, especially with wounding, causing cankers. So as we can see, Phytophthora can vary a lot in their life cycle, and it depends again on the host and the Phytophthora species. Now, there's several examples of Phytophthora in different scenarios. I would say that the poster child of Phytophthora is Phytophthora infestans, the causing agent of late blight. It's a worldwide distributed pathogen, and it's quite devastating in potato crops, tomatoes, and other solanaceae. We can also find here in California Phytophthora, diverse species of Phytophthora causing problems in orchards and either nut crops or prunes, causing root rots, crown rots. A common species that we were actually talking yesterday with a lab that works specifically on these kind of Phytophthora in orchards. Phytophthora niederhauserii is a common problem. We also have problems I like to mention Chile because I'm from Chile, and there's a problem with Phytophthora pinifolia that affects artificial plantations on Monterey pine, causing several tens of thousands of acres were affected in the mid early 2000s. There's also problems with Phytophthora cinnamomi in Australia. The Phytophthora cinnamomi is known as a biological bulldozer for its wide host range, affecting several species in Australia. So there's diverse species of Phytophthora causing different levels of aggressiveness severity depending on, again, on the host and the species of Phytophthora. But one of the big problems that we've been seeing and we're trying to resolve is how these Phytophthora get from one system to another, or how are they introduced into natural ecosystems? So I know it's wrong to point fingers, but one of the common factors is nurseries. Nurseries create a perfect scenario for one pathogen to jump to one host to another. Nurseries also apply abundant irrigation, which allow the plants to look healthy. A lot of fungicides are applied that not necessarily eliminate the fungi or pathogens, but normally suppress them. So once they're out in the open or without these fungicides, they tend to die or express symptoms quite severely. And due to this, to unknowingly knowing that different pathogens are in nursery plants, they can be transported. And one of the cases that's really known is the case of Phytophthora ramorum, the causal agent of sudden oak death here in California, where Phytophthora was first detected. This Phytophthora ramorum was first detected in Germany in a rhododendron plant, but it was later in the early 2000s, I think like around between 95 and 2000, it was then detected in California affecting tan oaks. However, once it was detected, it was said that the epidemic was already widespread. And then thanks to human movement of plants or plant trade, the pathogen was dispersed across great lengths across California. So we can see that these plants, moving plants at the same time, end up moving pathogens. So one of the routes from nursery to natural ecosystems is through restoration. So I'm going to explain a little bit how restoration is connected to this, but first I want to explain what is restoration. So restoration is a way that severely degraded ecosystems are restored. There's two main ways to do restoration: a passive restoration, where you normally control for weeds, control for fires, allowing a natural regeneration of different ecosystems, like reducing grazing, etc. Now there's a method that's called an active restoration, where you literally outplant nursery plants out into severely degraded areas to help restore the ecosystem. You transplant between 1 or 2 year old plants out into the open. There's other methods where people apply seeds, a slurry of seeds into a plant basin, a method called hydroseeding, and you just weed a little bit in the plant basin and then allow the establishment of these plants. However, as we just mentioned, nurseries are a place where many pathogens are local residents of nurseries are hanging out in those plants and inadvertently then transported from nurseries into these restoration areas. So pathogens unknowingly are being introduced into these natural areas, where you can see that once it's out in the open, it can easily move into adjacent areas. So this problem with restorations and nurseries is not a new problem. It's something that has been detected, and it's increasingly detected in California. One of the clear examples, one of the first examples, was the case of Phytophthora tentaculata, where it was first found in a nursery affecting monkey plants, monkey flower plants, I think it's called. And then years later, it was then found in restoration areas. Later, a group from Berkeley, Laura Sims, did a similar analysis where they sampled several restoration areas and nurseries and found several isolates of Phytophthora crassimiras. They were able to detect this Phytophthora. And they found that it was amongst these nurseries and restoration, it was common 4 different haplotypes of COX-1. And they found 2 haplotypes that were common between the nurseries and restoration areas. They also found that not only genetically they were connected, but also like phenotypically, because they found that isolates from both nursery and restoration areas exhibit a variation sensitivity towards metalaxyl, commonly used fungicide, which indicated that if there's a variability, it could indicate that they were previously exposed to these compounds. and the only place they can be previously exposed to is in nurseries. So they made that connection. And then after that, after noticing that there are several cases going on, especially in the Bay Area, more people started sampling more restoration areas and more nursery stock and started encountering several Phytophthora species. Recently, in this year, again, the group from Berkeley published another study indicating that Phytophthora are found in restoration areas and that can easily spread to adjacent areas where previously no Phytophthora was found. They sampled the same species in restoration areas and then farther away control areas where further away from restoration areas, and they found no Phytophthora associated with a certain plant species. So several studies are coming out that are starting to connect this pathway. So this brings me to the case study I wanted to talk to you about, and which is part of my dissertation topic, where in around 2016 to 17, again, just like what was happening in the Bay Areas, restoration areas were sampled in the Angeles National Forest. Also, nurseries associated with these restoration areas were sampled, and several Phytophthora species popped up. So, and this is why in 2017, with a collaborative project with the National Fish and Wildlife Foundation, the Forest Service, and us from UC Davis, collaborated on a project to detect Phytophthora species in the Angeles National Forest, specifically in areas that are planned for restoration, the areas that have been disturbed by anthropogenic reasons or fires, and we sampled for Phytophthora. So this is, I would say, the first part of my dissertation topic where we went out and sampled these pre-restoration areas. Well, a little bit about the Angeles. The Angeles National Forest is located in northern part of LA County. It's managed by the Forest Service and protected by the Forest Service, and it's considered part of a Mediterranean climate. And with that, it's considered one of the most fire-prone places in the United States. So it's a place that has severely been degraded by different disturbances. One of them, as I mentioned, was fire. And the project that we were working on and currently are also is fires that occurred about 15 to 20 years ago. Three main fires that affected over 20,000 acres: the copper, the copper, the Sayer, and the ranch fires. As we can see here in the map, the copper fire where we sampled the most is located in the northern eastern part of the Angeles. There's also a southern part of the Angeles National Forest where the Sayer fire occurred. And then there's another fire that we sampled that's called the ranch fire, that's more on the western side of the Angeles. So these areas, as like I said, burned about 15 years ago, and with fire severity and flooding and OHV tracks, what we refer to off-road vehicles, really degraded the area. So several restoration areas were initiated in those fire areas. And I always like to show this picture of an active restoration area and how intensive the labor is. I can see at least like 200 little holes there where they're going to actively outplant nursery stock. As we can see, that we increase the chances of introducing an unwanted pathogen. So we proposed two main questions. The first question is, what are the patterns related with distribution of Phytophthora species? And then finally, which is a very broad question, is how these oomycetes, in this case Phytophthora, potentially impact native plant communities. So as I mentioned, the copper fire area is composed of four main vegetation types, composed of a coastal sage shrub, where we can find a lot of Salvia mellifera, for example. There's a chamis chaparral, where we find, again, chamis, a little bit more drier ecosystem. Adenostoma fasciculatum is the main the main species there. And then we have another vegetation that's like a little bit of an oak mixed chaparral with scrub oaks and coast live oak, Quercus agrifolia. And then we have a lot of annual and exotic grasslands too covering the area. So what do we do? Between May 2018 and March 2019, we went out and sampled these pre-restoration areas, which were about 27 sites. At each site, we sampled 10 samples. We tried to aim for places that we probably would encounter Phytophthora, especially focusing on symptomatic plants. And with pears and rhododendron, we baited rhizophyre soil from underneath certain plants. And through ITS and morphology, we isolated and determined and determined different Phytophthora species. So this is the natural landscape that we tend to find in the Angeles, with again chaparral islands mixed with annual and exotic grasslands, and in the background beautiful power lines. And then inside the Angeles, we also find these riparian areas. We find active restoration areas. We also find off-road vehicle tracks and more disturbance, and also some pine some conifer stands, and basically mostly gray pine and big cone Douglas fir. Amongst the phytophthora that we encountered, the majority were Gonapodides, followed by Crasimira. And Crasimira, if we remember, was one of the species that was found from the Berkeley group in restoration areas in the Bay Area. We also found Cactorum and Multivora, other two commonly found Phytophthora species in restoration in nurseries. Sorry, we can see here the map of the copperfire area, where all the red dots indicate the positive sites and the whites indicate either Pythium or negative Phytophthora. Now, if we do a comparison with the different vegetation types and where we encounter different Phytophthora species, we can see that the oaklands harbored the larger diversity of Phytophthora species, and then Phytophthora crasimira was the one that was encountered in most of 4 out of the 5 vegetation types. So it's one of the most widespread Phytophthora that we encountered. Here are some associations with hosts. Again, Eridictium crassifolium was, we found some dead plants and where we isolated Phytophthora. Now it's kind of hard to find or to actually determine what is asymptomatic chaparral plants, because they tend to dry up and really die off some of the branches. So it's kind of hard to associate symptoms with actually symptoms of a natural physiology of the plant, or is it actually part of the pathogen? And that's what we kind of want to discern and narrow later on in our project. We found the majority of the Phytophthora species were associated with riparian zones that were passing through those vegetation classes, which does make a lot of sense regarding the biology of Phytophthora that requires a lot of water to cause infection. I'm gonna start going a little bit faster because I think my time is running up. But these are the common riparian areas that we find in the Angeles, where they almost act like it's an oasis, where you find riparian areas associated with a larger diversity of different vegetation. And then as you move out of these riparian areas, you start getting into grasslands or these chaparral islands. And then if we do an aerial picture of our detections, the blue dots indicate either negative or just Pythiums, and the red indicate Phytophthora, in this case, Phytophthora crasimira. And if you follow the line of the trees, it also follows the line of the riparian area. And then if you move and we see other sites, we see the same tendency, finding Phytophthora generally in the riparian area, but as you move out, less and less Phytophthora. And then we said there has to be some sort of a connection with the riparian areas, because in the Angeles, what we find a lot are what we call perennial and ephemeral rivers, either streams that are seasonally show up, or streams and rivers that are constantly with water all year long. And we found the Phytophthora species only associated or mostly associated with ephemeral and perennial streams, with the exception of one Phytophthora crasimira that we found on an off-road vehicle track. Okay. So then the next question, and a big broad question that we're trying to answer, is how does these phytophthora potentially impact native plant communities, which is not an easy question and is not something that can be answered in 1 or 2 years of studies. For example, Phytophthora morum, we've been, in our lab especially, has been studying this pathogen for over 20 years, and there's still a lot of answers that we still need to obtain. So as a preliminary way to assess this is through pathogenesis tests. And the way that we did this, we selected 5 main species that we were interested in and that were commonly dominant in each of these vegetation classes. We chose chamis, thick leaf yerba santa, California buckwheat, black sage, and coast live oak or Quercus agrifolia. And we're trying 2 different inoculation methods through soil infestation and soil spore inoculations. The soil infestation is actually quite a neat way to do this. We make these spawn bags with vermiculite and oat seeds and some V8 juice, and we inoculate, we sterilize that bag, and then we inoculate with Phytophthora. And then this mix is what we use then to inoculate an adjacent area to the potted plant. And then to create conducive conditions for Phytophthora to initiate infection, these plants undergo a 72-hour flooding period, which creates the formation of sporangia and spores, as we know, and cause infection in plants. These trials take a while, so right now I'm just going to show some of our preliminary results. This is, I believe, between 1 and 2 months into the inoculation. We already see that different phytophthora are causing different symptoms on each of the plants. A lot of wilting we see, then we see some, especially in the bottom leaves, some leaf necrosis, and then cankers forming on the stem. But once we process these, and once we process these, we already processed them, we get to see different aggressiveness on the root system too, causing different levels of root necrosis or root rots. And a way that to discern different aggressiveness is also through root weight. So yeah. And then another way that we're doing these inoculations is through sow spores. This is Liz, one of our undergrads. She's helping me out with a lot of these inoculations. And we're trying this neat new method of putting the little seedlings inside of a scintillation vial and then flooding these seedlings with sow spores. And obviously the controls with distilled water. This is what after all the plants have been inoculated kind of look like. And then I wanted to show a few examples, especially with Phytophthora cactorum, how through this method we can easily follow the progression of disease. In 2 weeks, Phytophthora cactorum was quite aggressive on the roots, and in some cases moved up the stem and caused a necrosis on the stem, and then a whole wilting of the plant. And here we can compare with the controls. This is a good method to test out also intra and interspecific viability of these Phytophthora species. In a very reduced and a very reduced area, we can test out several plants. And then I kind of want to talk a little bit about like what kind of things can be done by analyzing these kind of Phytophthora distribution and diversity of species. There's a group from Sweden and then a group of Spanish researchers that are working on how different functional traits are allowing certain Phytophthora to be less or more invasive, and how different environmental filters reduce or increase the species richness depending on the scenario these are encountered. From nurseries where we find a larger species richness, and then there's a environmental filter, a selection that reduces the amount of species that we find eventually in natural areas. There's also a group from Australia that's working on analyzing distribution of Phytophthora and how either local or exotic Phytophthora species are affected by elevation or different environmental factors. And they've seen that native Phytophthora species are a lot more affected by these different types of disturbances, especially in this case, they did with elevation, and how exotic species are not as affected by elevation. In California, also, I want to talk a little bit about the work our postdoc is doing, Tyler Burrett. And I collaborated a lot with him in this project where we did a compilation of all about 800 ITS sequences of Phytophthora species associated with California. And we separated them in 4 different scenarios or contexts, from natural forests, in nursery horticulture, in ag land, and then in restoration areas. And this Venn diagram indicates how there's certain taxas that are unique to each of these areas, but also taxas that are common between these different contexts. And discerning out these kind of relationships really help us to determine, and especially compiling a lot of these environmental factors, what areas are potentially conducive for Phytophthora and what kind of Phytophthora are we expected to find. And I didn't want to just paint bad news about Phytophthora restoration areas. Susan Frankel last year published a really interesting article where they mentioned the different things that are being done to reduce the potential risk of introduction of Phytophthora, from soil solarization to specially built nurseries that avoid the introduction of Phytophthora into the nurseries, and the introduction to natural adjacent areas. And also the Presidio Nursery in the Golden Gate National Park in San Francisco in the Bay Area, they're doing a lot of Phytophthora testing using the same method that I was using to detect Phytophthora in the Angeles National Forest. So there's a lot of things that are being done to prevent these kind of situations. And the last I wanted to, before I finish, is that there's a new set of proposals that I actually do next week. And we're gonna present a proposal to narrow down more of these distribution type related questions regarding Phytophthora, especially the relationship with ephemeral and perennial rivers, to determine the likelihood of encountering Phytophthora at different distances. This could definitely help restoration managers to know which areas are more conducive or less conducive for Phytophthora. And then I want to finish with some acknowledgement, especially everybody from the Rizzo lab, and Susan Franklin, Dave Rizzo, and Tyler, who really have helped me work through a lot of these questions and organize a lot of these data, and in general help with everybody from the Rizzo lab too. So thank you so much for allowing me to talk about this topic. So thank you.
Awesome. Well, thank you. Sebastian, you kind of ran a little late, so
Yeah, sorry.
we're gonna not get into the questions, but if people have questions, you're welcome to answer them through the message chat option. And then we'll just go ahead and start with Lisa, okay?
Yeah.
Yeah, so you're welcome to stop sharing your screen.
All right, thank you.
Thank you. Alrighty, so thank you so much, David. Excuse me. Thank you so much, Sebastian. And so questions will be hopefully answered below. But our next speaker is Lisa, and here's just a little bit about her. She's a PhD candidate and NSF Graduate Research Fellow in Ecology at UC Davis, working with Dr. Dave Rizzo. In her dissertation, she studies artificial plant mesocosm communities and large forests by monitoring their networks infested with sudden oak death, to characterize some of the mechanisms underlying the diversity disease relationship in natural ecosystems. She also studies long-term changes to canopy gaps in Yosemite Valley to describe the many interacting abiotic and biotic forest disturbances. Lisa, you're welcome to begin.
Thank you. I'll unmute myself. Let's see, PowerPoint. Yeah. Okay, so let me first collapse this so that I don't get distracted. Okay, so today I'll be presenting on essentially half of my dissertation, which focuses on, or that finds that the direction and drivers of the diversity disease relationship are distinct across hierarchical levels. And this project was done in collaboration with Alison Simler Williamson, who's now at Boise State, as well as Dave Rizzo. And I'd also like to acknowledge my funding agencies, the NSF GRFP, as well as my graduate group in ecology at Davis. So as many of us know, biodiversity loss is accelerating worldwide, and this loss of native species and biological homogenization often has negative impacts on ecosystem services that we depend on. So whether that's carbon sequestration or productivity or erosion control, biodiversity loss has a negative impact. But when it comes to disease, the verdict is still kind of up in the air. So it's true, a lot of times biodiversity does limit disease risk. And so this negative relationship between diversity and disease is called the dilution effect. But sometimes increasing diversity doesn't actually help reduce disease, it can actually amplify it. And so we call this negative, or we call this positive relationship, the amplification effect. And so over the last decade, there's been a lot of back and forth about whether the dilution effect is general or not. And it's caused a really big rift in the disease ecology world. So much that even commentaries need to come out calling everyone to cool the temperatures. Let's diffuse the dilution effect debate. So the most productive path forward is to actually ask, what are the underlying mechanisms driving the diversity disease relationship? Okay, so besides just addressing kind of this rift in the academic world, it has real world applications, because understanding how diversity in nature affects disease dynamics can affect how we manage our forests and how we manage our natural lands. So if diversity predictably co-varies with fewer reservoir hosts that are the things that they're the hosts that spread disease, then conservation of biodiversity could be a viable win-win strategy. You save biodiversity and you limit disease. But if that's not the case, if say reservoir hosts don't correlate with diversity, then we need targeted management of specific species so that we knock those out and we control disease outbreaks. So I'm going to address that overarching question about mechanisms driving the diversity disease relationship using the sudden oak death system in the Big Sur region. So what you're looking at right now is a landscape in Big Sur modeled with a whole bunch of dead tan oak and oak trees. Okay, all of these dead trees. And this disease is caused by Phytophthora morm, which is yet another Phytophthora species, which is an invasive species that first was noticed here in 1995. And since then, it's estimated that about at least 48 million stems of tan oak and oak trees have been killed along the coast of California and southwestern Oregon, although most of that mortality really is in California. Okay, so this is a super ecologically devastating disease. And the way that this works is most of the spores, most of the sporangia, are produced on the tips of these bay laurel leaves. And bay laurel itself is not negatively impacted by the pathogen, but it's so good at spreading disease, or so good at spreading spores. So we would call that it's highly competent, okay? And host competence here is defined as the ability to get infected and transmit a pathogen. So when these sporangia fly off the leaves of bay laurel, sometimes they land on oaks and tan oaks. Oaks and tan oaks both develop these lethal cankers, that will ultimately girdle the tree and kill them. True oaks themselves are not very good at producing inoculum, but tan oak can actually produce sporangia on its leaves and can also contribute to overall inoculum load, but definitely not near to the capacity that bay laurel can. And of course, there's other species in these forests. So, the role that they play in the epidemiological story in sudden oak death is kind of up in the air, whether they actually also contribute to overall inoculum load or actually get in the way of these dispersal pathways is unclear. Another thing about this system is about a decade ago, a paper came out led by led by Sarah Haas that showed that community-wide infection prevalence decreased with diversity. Okay, so now we're setting up the stage where there's a previously identified dilution effect. And so now I can ask this question, what are the underlying mechanisms driving this dilution effect? Okay, so I'm going to try out a new concept figure. This paper is in revision right now, and this will be the first time that I try it out. So let's imagine that you're looking at a low diversity community. Okay, it's filled with 3 types of species. One that is it's highly competent and it shows symptoms commonly. So you can kind of think about it. So highly susceptible and highly competent. And you can kind of just picture in your mind, maybe this is a bay laurel, okay? It spreads lots of disease. Another species that's in this low diversity community is these red circles, which are low competence, but also exhibit lots of symptoms frequently. And then another type of species, say there's potentially high competence hosts, but they don't show symptoms frequently. And maybe redwood or other less, redwood or madrone or whatever might be this sort of high competent, rarely symptomatic host, or maybe they might be low competence, rarely symptomatic host. Like I said on the previous slide, we just don't really know what they're doing. Okay, so because I don't know what they're doing, I'm just gonna leave this little redwood leaf in between those two. Okay, so let's pretend that like this low diversity community had low competence, rarely symptomatic species added to them. In this higher diversity community now, there's a couple of scenarios that can happen. In this high diversity community, let's just assume that these low competence, rarely symptomatic species reduced the density of competent hosts. Okay, so when you look at this high diversity community, all of a sudden there are fewer of these highly competent hosts. So if you count up the inoculum pressure from these red species with the halos and these blue species with the halos, there are fewer of them compared to the low diversity community. So when you look at individual level disease risk, which you might calculate with a Bernoulli model, then it decreases across the diversity gradient. At the same time, when you look at community level disease risk, which is, you can think of it as the number of infected plants divided by the total number of plants. So it's really sensitive to the proportion of symptomatic species. It also decreases across this diversity gradient. Okay, so at both the individual and community level, you see a dilution effect. Another scenario that might happen is the addition of these low competence, rarely symptomatic species is that, okay, it doesn't actually change the density of competent hosts, but what it does is it reduces the transmission potential of competent species in that community. So I show this by limiting the size of the halos. And so similar to the last scenario, individual level disease risk declines across the diversity gradient. And so does community level disease risk, because again, there's a lower relative proportion of symptomatic species in high diversity communities. In the last scenario, we can have a situation where adding these species doesn't actually change the density of competent hosts, nor does it change kind of the transmission potential of competent species. And so there's no plant-to-plant interactions. This creates no change in individual level disease risk, but still at the community level, there's a decline in disease risk because there's a lower relative proportion of symptomatic species. So I'm drilling this home because this is super important to understand my analysis. Individual level disease risk is sensitive to the absolute inoculum pressure, whereas community level disease risk is sensitive to the proportion of commonly symptomatic species. Okay, so this is tracking transmission potential. This is tracking essentially relative proportions of symptomatic species. Okay, so what I did in this project was I tested which one of these mechanisms or which one of these scenarios is occurring in the Big Sur system. So in order to do that, I'm using field collected data from 151 500-meter square plots along the region, this Big Sur region here in California. And they're all culture positive for Phytophthora morm. Okay, so within each of these plots, we collected plant community data as well as disease symptom data on all stems that were above 1 centimeter DBH. And in total, my data set includes nearly 5,800 trees, and each of these trees have multiple stems. So it's quite a lot of data. And most of the symptoms were found on 4 commonly symptomatic species. So that included bay laurel, where nearly 90% of the bay laurels that were included did show infections. And then also tan oak, where about 50% of the individuals were infected, coast live oak and shreve oak, where infection prevalence was much lower. And then there were, I believe, 16 or 14 other rarely symptomatic species, or there are 14 other species in this data set, and they were very rarely symptomatic. I believe only 8 out of many redwoods were infected, and only 1 maple was infected. Okay, so what's key here is that there were 4 very symptomatic species, and then the rest were very rarely symptomatic. I also looked at how the densities of those 2 key epidemiologically important hosts changed over diversity. So I did this, I looked at the densities of tan oak and bay laurel using a hurdle model. And so what a hurdle model does is it asks essentially 2 questions. The first one is how does the occurrence of the species change across diversity? And then if it's present, how does its density change across diversity? So the occurrence of tan oak didn't change too much across diversity, whereas bay laurel increased in occurrence as diversity increased. And then when the species was present, the density of tan oak also didn't change very much across diversity. And then bay laurel declined very weakly across diversity. So in total, when you kind of take into account these 2 separate processes, the overall change in the density of either species didn't really change much. Okay, I also looked at the number of plants of highly symptomatic species and minimally symptomatic species. And so this is important because it gives me an idea of the relative proportion of symptomatic species across the diversity gradient, which I repeated over and over again is really important for understanding how community-wide infection risk changes. So what I see is that the number of minimally susceptible species increased across richness in both forest types. I forgot to say that the colors are forest types, redwood and mixed evergreen. Whereas the number of highly susceptible species didn't really change much across diversity. And so what this does is the relative densities of these commonly symptomatic hosts decreases across diversity. Okay, another thing that I did with this project was I augmented the field collected data with laboratory sporulation assays. And so what we did was we collected leaves from 32 plants per species for the top 13 most common species in the Big Sur region. So you can see here Sebastian, as well as Carrie collecting it looks like some poison oak leaves. We brought them back to the lab. We incubated leaves. We quantified sporulation after incubating them. And I did that by scraping off these solutions from the leaves and counting sporangia under the microscope. And then I estimated sporulation potential, and I'm using these sporulation potentials in order to approximate competence, host competence. And I'm interested in host competence because if you multiply each species competence times its respective density in each plot, and then you sum that up across all of the species in the plot, then you can calculate an aggregate metric of you can essentially calculate net reservoir competence of the community. And so I'm going to call that community competence. And you can learn more about this in an in-press paper in plant disease. Okay, so in order to, I also looked at, so I was interested in calculating community competence across diversity because if higher diversity communities have lower community competence, then it would suggest there's a lower inoculum pressure. And then that might explain why a dilution effect is happening. I show all of this other stuff, including how communities are nested and also how, and I also show competence in order of ranked abundance of ubiquity, because it's grounded in theoretical, it's grounded in how, it gives us an idea of how community disassembly might affect disease dynamics. It's a lot more than I really want to talk about right now. So I'm just going to show it to you. And if you have questions about this later, then I can answer them. Okay, but the most important thing is that community competence, at least using these laboratory assays, it decreased across diversity. Okay, so my largest question in this analysis is, how does support for the dilution effect and the underlying drivers differ at the individual versus community level? And by answering that question, then I can get back to that original concept figure and synthesize which of the 3 different scenarios I think is happening. So I addressed this question using 3 models. In the first model, I assessed plot level infection prevalence using all of the host species. And this is the response variable. I do that using a beta binomial model. This looks kind of intimidating, but it's really, it's a binomial model, and the beta part is used for capturing extra dispersion. Okay, and so in this model, I include plot level covariates to control for other confounding variables like abiotic and other biotic confounding variables that might also impact disease risk. And I contrasted 3 competing models. So in the first model, I included a model that just had richness. In my second model, I included richness as well as the density of bay laurel and tan oak in order to understand what role that those species might have in disease. And then I also contrasted a model that had richness as well as community competence. And so the difference between model 2 and model 3 is that community competence not only incorporates the inoculum pressure from these 2 important species, but also from all the other species in the community. In this, I ran another model that essentially did the same exact thing that I just explained, except it's using a restricted data set. So it's just including these these commonly symptomatic species. And the reason that I did that was so that I could isolate the effects that rarely symptomatic species have on altering community-wide infection prevalence. And so, like I said, I ran the same models and I contrasted the same 3 competing models. And in my last set of models, I analyzed individual level infection risk only using commonly symptomatic species because, practically speaking, those are the only ones that actually showed infections. Okay, so in order to do that, I ran a Bernoulli model. It has lots of terms in it where some of the terms address individual level variation, species level variation, as well as plot level variation. Within this plot level term, I included the same exact plot level covariates that I previously explained, and again, contrasted 3 competing models. Okay, so what I'm going to show you is coefficient plots, and when an estimate lies on the left side, it means that it has a negative impact on the response variable shown up here. If it lies on the right side, it has a positive impact, and when it crosses this line, this one-to-one line, it means that it has a probabilistically unimportant effect. Okay, so on its own, richness had a negative effect on plot level infection prevalence when all host species were included in the data set. And so this jives with the previous analysis from Haas et al. it would suggest that there's a dilution effect. Okay, and including these extra variables for beta laurel and tan oak doesn't change the effect of richness. And so it suggests, okay, the dilution effect is still occurring even after accounting for the density of these important species. And when you include community competence into the model, it looks like richness is no longer important. And so that might suggest, well, hey, extra species are important for explaining this dilution effect. Hold that thought. When you exclude those rarely symptomatic species, the effect of richness is no longer important, and so the dilution effect disappears. And that doesn't change when you include these other important species, nor does it change when you include community competence. And so my interpretation of this is that it's actually those minimally susceptible species altering the proportion of symptomatic host across the diversity gradient. That's what's causing this dilution effect. And then when you look at individual level infection risk, you get very similar results to panel B. So there's no dilution effect, or the effect of richness is not important at the individual level. And it doesn't change when you include other covariates. And what's constant across all three models is that the effect of beta laurel is very strong, overwhelmingly strong, especially compared to other species. So tan oak is not important in spreading infections, and community competence, yeah, it does have a positive effect on disease, but it's much smaller than beta laurel. And probably the only reason it even has a positive effect is because beta laurel is integrated into this variable. So let's go over what just happened. Transmission risk is driven by the density of a single highly competent and abundant host, beta laurel. There's also a higher number of rarely symptomatic species in diverse communities, and these numerically dilute the proportion of infected plants in the plot, but without affecting transmission risk. And so when you bring it back to that concept figure, what's happening is these other rarely symptomatic species are actually very low competence hosts, and they don't have any impact on transmission risk. They kind of just sit there, creating no effect on the individual level disease risk across diversity, but at the community level, there's still a decline in disease risk because, again, there's a lower proportion of susceptible hosts. So richness doesn't limit the risk of an individual acquiring infections, but richness does limit the percent infected plants in the community. So the takeaway is the direction and drivers of the diversity disease patterns are distinct across hierarchical levels. And this matters because if your goal is to protect specific species, say like the tan oak, which is culturally and ecologically important, then saving biodiversity won't help, and you need targeted management of beta laurel. Let me preface this, that these management implications are specific to the Big Sur region. If the goal alternatively is to protect ecosystem resilience, and you might measure that in percent infected of the forest, then yeah, sure, maybe saving biodiversity could help because it does correlate with a lower relative density of infectious and susceptible species. So with that, I'll take questions, and yeah, what's there?
Wow, awesome. Thank you so much, Lisa. I see, Charlie, you have a question. Would you like to read it, or I can read it for you? Okay, maybe I can read it. All right. So what level of Phytophthora disease did you see, or would you estimate was on the coast live oak?
Just based off of raw numbers, I think the value is about 12%. Yeah, much lower than tan oak and bay laurel.
Interesting. Cool. Do we have any other questions?
Can I just, I guess, say it out loud?
Absolutely.
Thank you so much, Lisa. That was really incredible. Do you have any hypotheses for what kind of differences could see in other regions? You mentioned that this is specific to the Big Sur region. Would you expect to see sort of different conclusions in other regions, or the same, or?
Oh, yeah, I mean, so I wanted to preface that because of course there's so much variation in this disease system. Yeah, so I mean, I wanted to preface that because I'm particularly thinking about Oregon, where they don't really have bay laurels, and yet they still have spread of this disease. And the results from this analysis says that it's bay laurel that's spreading infections, not really tan oak. But of course, that can't be the case up in Oregon because it's primarily spread by tan oak. So I just wanted to preface that because these conclusions certainly will not hold true up in Oregon. And I don't know, maybe they also won't hold true in like the northern part of California, where saving tan oaks is super important up there because they're very culturally important for many of the tribes. And obviously Northern California, Del Norte County is very close to Oregon. So I wanted to preface that.
Thank you.
Interesting. Tyler mentions that they do have bay laurel in Oregon. It just isn't susceptible to P. ramorum up there for some reason.
Yeah, those Oregon myrtles.
Yeah. All right. Well, we've hit 5 o'clock. If there are any other questions, Lisa did put up her Twitter account. Sebastian, I didn't see if you had a Twitter or not, but hopefully through that contact, people can ask you guys questions later on. But thank you again for joining us, and very excited to see you all next month for our next set of speakers. Okay.
Thank you.
Thank you again to Lisa and Sebastian. Wonderful talk.